Welcome back, and today we're going to talk about autism, the neuroscience of autism. So, hopefully we have a general idea of what autism is. It's a sort of social disorder that has a bunch of different hallmarks. So how do we diagnose autism? It's generally considered to be a neurodevelopmental disorder, so that's important of when it shows up. Tends to show up early in development, starting around, well, some people may say as early as a year you can start to see things, but a year and a half, 2 years is the real time where individuals may start to be diagnosed, but it can also be diagnosed a bit later depending upon severity. But a key thing is that people with autism at birth seem completely normal, and they seem to develop initially normal, and then something along the developmental path seems to go astray. Behaviorally, we diagnosed it. It's a behavioral defect, right? And about 1 in 59 children born today are thought to have it, more boys than girls in general. A big hallmark of this is a deficit in social communication and desire to sort of communicate with others. Again, when we talk about Alzheimer's, there's a bit of a withdrawn effect to that, and that's sort of similar here as well. So it's both a desire to communicate and the decreased ability to communicate and interact with others as is considered normal. Deficits in interactions across multiple contexts, meaning that they have hard sort of switching from one task to another, just sort of as they need to. Repetitive behaviors and interests. So this is, I think, a very stereotypical one, but it is true. They may want to keep doing the same thing over and over again, play the same game, listen to the same music, watch the same movie. And that's true of a lot of kids, but there's a line that gets crossed at some point and that continues throughout the rest of their life. They also may have very specific interests that they really bore down in, and that they know super well, and that's all they want. Generally diagnosed earlier, around 3 to 5, with other developmental delays. Also, when we talk about autism, and I'm not going to say I'm a clinical psychologist to really dive deep into this at this point, but autism is considered now to exist as part of a spectrum of disease. So ASD is autism spectrum disorder. So you can have more milder forms of it. For example, Asperger's is typically what we call individuals who generally are able to get through life in a normal fashion, but they do have some, though they impact their lives for certain, they have some social defects. Often later, people with Asperger's, they're not at the severity as somebody with sort of more severe autism, where they are not. Usually people with Asperger's are able to function independently and hold down jobs for the most part. So given all of this, what we know about autism, how would you study this in an animal? And if we think back to talking about reductionism, this is a disease that I think is hard to reduce down to an animal model, like a mouse. What does a social deficit look like in a mouse that is relevant to a human? We behave quite differently, right? So going up and sticking your nose in like somebody you don't know, that's normal mouse behavior, and if they don't do that inappropriately, does that mean that is showing some kind of autism? It's really a tough call to say. So because it's a developmental disorder, so there are lots of reasons to think this, both the timing of it is key to that, but we do have some evidence here where we're looking at a particular part in the cortex, the neocortex, frontal cortex of individuals with autism, where normally when we talk about this part of the cortex should form layers or lamina, right? And that's what you're trying to see here. If you look, you can see sort of a bluish cyan, an orange, a yellow, a purple. And these layers are very stereotypical all throughout the brain. And they're labeled down here too. They give names 1, 2, 3, 4, 5, 6, 7. But the key to here is, like, if you look in an autistic individual, they can find examples of people in this New England Journal of Medicine article who have gaps where these layers break down. And there's still cells here. It's not just a loss of brain tissue, but they seem to not have formed the correct structure. This is very much a hallmark of a neurodevelopmental deficit because we know these layers form quite early, and they also happen to form right around those earliest times that we can sort of start to diagnose it. We're looking shortly after birth up to about 2 years when these layers should be pretty well formed. So that's an insight we have into the fact that this is a neurodevelopmental disorder and what is going on. Now, as to why this is going wrong, I think we have some to learn, but we have some ideas. One of the key things it points to is genetics. So we are not at a capacity, both as everyone, but particularly as this class, to really dive deep into the genetics of autism because it is complicated and we don't have the answers yet. But one of the big answers is that it's complicated. And so how do we know it's genetic? Or at least partially genetic. So if you have twins, right, 70 to 90% chance that one twin has autism, the other one does as well. And this is the same as if you have identical twins and they are separated at birth. So that sort of takes out the impact of environment. So that says something, that clearly there has to be something about genetic background. It also says it's not purely genetic background. There's some factor of environment, right? If we come back to nature versus nurture, this seems to be more nature than nurture, but there's some nurture. The thing to point out here, early on when they started diagnosing autism, one of the key things they thought it was all nurture, right? These kids aren't developing correctly, and who got blamed? The mothers got blamed. You were being a cold mother, so they didn't want to, interact socially. One of the key things a lot of autistic children do is they don't like to be hugged or to give hugs, so they blamed it on the mother, which is a terrible thing to have done. And we know it's completely not true now. When I'm saying environment, it's a more complex environment than that. We don't, again, know the answers, but could it be sort of what you eat, your chemicals you're exposed to, different things like that are likely to have had some impact, but we don't understand. So let's get back to genetics. We know there's a high percentage of this disease is caused by genetics, but it's complicated. We haven't really found, and I'll talk about a couple cases of autism-like disorders that do seem to be due to one gene being wrong. But the vast, vast majority, we know it has to be multiple combinations of genes in a way that's sort of, we aren't good at predicting yet, right? And you can kind of imagine the way this would work is that maybe, as humans, we have about 23,000 genes, right? And the way we could look at it is maybe there's about 400 of those. I'm sort of pulling a number out of here. This number changes a lot. Genes that are relevant to autism, and it could sort of vary, but if you had the wrong version of, say, 35 of those genes, maybe that would be autism. Whereas if you only had the wrong version of 34 of those, you'd be okay, right? And if you had 36, maybe it's more severe. But it does matter which of the 35 you have. So maybe some of those would be okay, but maybe if you have a really bad version of one, then you only need 20. That's what I'm saying. It's complicated and we don't completely understand, but we do know some types of genes that seem to be important. Another thought about how autism, what's the functional, the underlying autism. We talked about the idea that the different cortical layers might be a problem. Another one, and this goes along with the development, is the idea that there are too many synapses. And this comes up a lot. Too many synapses sounds like it might be a good thing. How can you have too many synapses? Aren't synapses good? But as we've talked about before, as you go across development, you see sort of early on, we make a whole bunch of synapses. And then we spend the rest of our life sort of pruning them, right? Kind of like I always imagine like a bonsai tree, right? Removing is sort of creating this more elegant, more pristine structure that is more functional. And that's what our brains do. And it's important to remove these synapses. And we'll talk about this a little bit more. Talk about schizophrenia too. It seems to be a problem. And so if you're looking over here, this is that idea where this black line is looking at multiple individuals and seeing how in a certain part of the brain, how many synapses there are per distance, right? And if you look across age, this number should go down. That's healthy. That's normal. But if you look at people with autism spectrum disorder, it does go down, but at a much slower rate. So this is one of the first real strong cases to be made that there are too many synapses, and that seems to be Related, and we have some, if you dive deeper into the we do have some more functional understanding about how this could be, but there's still no real proof to say exactly why this causes autism, or is there a way that we can simply remove them and fix autism? It's more complicated than that, but this is clearly part of it. If we can understand this, the key is that if we want to cure autism, we're probably going to have to do it at this age, right? Where we can sort of get somebody back on track to decrease as they should normally. Once they get to here, it's probably hard in your 20s to start removing synapses safely. So this is, I always make sure to talk about this a little bit, and I know it's a controversial topic, and I don't want to go too deep into that. I just want to tell you sort of what I know, and we'll leave it at that. And again, with the topic of vaccines is maybe never more relevant than when I talked about this. But, so there's the idea that vaccines cause autism. And I would say as a scientist, I've personally looked into this and I've talked to other people, and there really isn't any good basis in this. But let me talk a little bit more. So where did this come from? There was one guy, I'm not even going to say his name, from England, who he was a medical doctor, and he published one study saying there's a link between sort of groups that were given a vaccine and those who are not in the group with the vaccine ended up having higher rates of autism, right? That paper is, he happened to also be selling vaccine alternatives. So there's a sort of conflict of interest, something that we actually control for quite carefully now in the U.S. and have for years. It would be very hard for him to have got funding for the research like that. You can't sort of do research just to push your own agenda now. So that paper has since been retracted, but people don't realize that. That isn't, or they think it's sort of a conspiracy type thing. And he's also had his medical license revoked. So here's the thing, though. This idea that autism could be caused by vaccine, there was at one point a reasonable hypothesis there. You could get there, right? We know it's a developmental disorder, and what happens a lot in those early years, you're giving vaccines. And we know that there is some interplay between the immune system and the nervous system, and we'll talk about this again next time, where we know that the immune system is actually important for removing extra synapses. So that made sense, and it was a good hypothesis to start with. But the problem is that the experiment to test it was not good. So a lot of other people, besides this guy, and before and after this guy, have looked into it. And they did a much better job controlling for a number of different things. He sort of cherry-picked data to get his hypothesis. If you really control to get a representative sample of populations, and I said, and I think this number is way higher now, but greater than 30 other publications have been published showing absolutely no link between autism and vaccines. The most recent study, and his study, I don't want to misquote it, but it was relatively small. Most recent study that I found, and this was last year, so I haven't actually updated it for this year, was looking at 650,000 children and found no link. Now, we've talked a lot about how doing human studies is complicated, and usually it's hard to get enough numbers, and we're talking about then getting like 10 or 20, right? Maybe if you're lucky, a couple hundred. So looking at 650,000 or more than 650,000, you do at that point clearly have enough power to make strong statistical significance. And even if there had been a tiny link between them, at this number, you would have found it. And they didn't. And I would just want to say, in my mind, this is, scientists can get a bad rap. And a lot of people think that there's some link between scientists, trying to get money for drug companies, and I think maybe even at one point I would have believed that. And I'm not going to say there aren't bad scientists out there who are like that, but that is such a small percentage of people, and this is sort of horrifying to most scientists. The idea that you would do this, and really, your career as a scientist is done once you try and do one of these things. Even if there's a whiff of something like this. remember a lab in grad school where they had something that was actually supposed to help restore. It was something with the immune system. It was that was they had a drug that they thought was going to be very helpful for sort of boosting the immune system. And there was one person in the lab, not even the head of the lab, who was actually sort of altering other people's results by putting chemicals in some of their reagents. And the lab caught this. They caught this guy eventually, and then they retracted all the papers that may have had anything done with it. But that was it, right? Like, people who had done good work, but because they were associated with it, they never really had a career in science before or after that, right? Like, just scientists don't do things like this very often, and they're sort of hypercritical of themselves. So really thinking that somebody in science is doing this, it seems crazy. That was a tangent I didn't expect to go on. All right, so let's get back to the genes involved in autism. And if we're linking this to the synapse, so this is a picture of a synapse, and then it shows all the different proteins, right? The proteins are what genes make, that are thought to be linked to autism. And we can see there's a huge number of them there, and don't worry about it. But the thing is that this is where almost all the genes are. They make stuff that's at the synapse. So it makes sense. There's something about autism in the synapse, but exactly how this all works together and what misregulation leads to autism, we really don't know. Right, so this is something to think. Why do you think genetics for autism is so complicated compared to, if we think about something like cancer, usually we're talking about a couple genes at most. It might even, cancer quite often is just one gene that causes it going wrong. But autism, we're talking about dozens. All right, so there are a few. They aren't quite autism. I think they would generally be considered sort of autism-like diseases that do seem to have clear genetic links. Fragile X syndrome is one of them. If you remember back to the paper that we read on autistic individuals, they said this is something they screen people out for because it's a pretty severe form. It's a genetic form of autism, change in the X chromosome, which makes sense why males are usually more affected than females, because usually females have one good copy still, whereas males would have only one bad copy, and that's the only copy they have. The key gene that is affected is FMRP. Without it, excess proteins are produced, inhibiting protein synthesis, ameliorates symptoms in mouse models. So what does it mean? Again, this is a sort of generic effect where you have a mutation and it just makes more of a lot of different proteins. That's not really specific enough to completely understand the disease, but we do know that if you knock it back down in a mouse, at least, they seem to restore back to something closer to the control condition. Right. Another example is Rett syndrome. This is also X-linked, but it only occurs in females. And the thing is here is that this is so much severe that you basically have to have one good copy of the X chromosome to even survive this. So males who are developing with this mutation even before birth actually die because males can only have one copy of it. This is due to a mutation in the MECP2 gene. I just sort of told the answer why it occurs in females. A range of developmental defects. It's not necessarily considered autism, but it clearly has autism-like phenotypes, right? And another key thing is here is it seems to be, this is showing changes in gene regulation by size. So don't worry about it too much, but know that bigger genes are over here on the right, smaller genes on the left, and sort of anything pretty close to this bar is normal. So this is looking at the difference between mice that have MECP2 and those that don't. And so pretty normal until you get to the really big genes, and then something is off, right? They start making more really big genes. This gets back to the role of FMRP that we just talked about, and Fragile X as well. We don't know why this, again, causes this disease, but a key thing is here is that for whatever reason, these big genes tend to be genes that are only used in neurons. The general thought there could be something like it takes a lot of energy to produce bigger genes, and so most cells get turned over all the time. But your neurons don't, right? You get the neurons you have for life. So here you can make these big genes, and since it's going to stay around a while, it might be worth the energy to do it. All right. This is one last thing. I'm always a little hesitant to talk about this, and I do try and look this up. I looked it up last semester. This was sort of the state of where we're at. And again, I'm a real careful territory, but this looks to be a pretty good study that makes me think something's here, but I would still wait for sort of more data, more experimentation. This is the idea of looking at the gastrointestinal aspect of autism. Something that's been hypothesized for a while. Individuals with autism also have sort of a lot of stomach ailments, right? Often abdominal pain, constipation, diarrhea are associated with them. Never completely understood why. And with talking more about the microbiome, basically the bacteria that you have in your gut seems to have wide-ranging effects. We know it definitely affects things like mood and other things. So this concept of the gut-brain axis has come up. How those link, how they affect one another. You can kind of go through the data here, but so what they did here is something, I don't know if you're familiar with this concept or not, but basically, the first time I heard it, it was called a transputation. Back in grad school, I don't know if anybody really uses that term anymore, but the idea was that if your microbiome is wrong, you've got the wrong sort of bacteria in your gut. Basically, if you take feces from a person with good microbiome and sort of put it into you and use it to replace your microbiome, maybe you can, get back to normal. And this works in some cases. There are different diseases that it's being looked for, gastrointestinal normally, and if you have sort of chronic C. diff infection, it's actually approved for this, and there are people who can, buy this medicinally through your hospital, of course, and they do a procedure. It works for that. It works quite effectively. If you have chronic C. diff, you get one of these procedures, and you go back to normal largely. And it's been looking at for a lot of different gastrointestinal diseases like Crohn's and other things. Now, here's the thing. They basically did this treatment on autistic individuals and tried to look, did this have an effect on them? And that's what they're showing here. These are basically, I'm trying to look at these quick here, all different ways to measure sort of behavioral defects in autism. And you can dive into the paper to find out the details of all these different tests. But the key thing here is it's always baseline, so that's what they were before anything, treatment, 8 weeks after treatment. And the big thing about this paper is it goes all the way up to 2 years after treatment. And you are seeing enhancement that persists, or in some cases even continues to improve out to 2 years, which is a huge deal. And now these are also another thing that always makes this a little believable. A lot of these are not so huge differences that you would think like, oh, this is like a crazy sort of cure-all thing. But they are sort of big differences in the field where you don't really have a lot of ways to treat besides sort of behavioral treatment. And again, I think this needs more research, but I think it's exciting. One thing I always wonder about is how did this happen in the first place? If it's a neurodevelopmental disease, when did the gut biome sort of go wrong in the first place, and how does that actually cause it? We've got a lot to learn here. All right, to summarize what we learned, we have lots of good ideas about what causes autism, but really understanding that mechanism to go from a set of genes or a particular biological phenomenon to actually understanding how that causes the behavioral defects in autism, we are still connecting those strong answers, right? If it was easy to answer, we'd probably have treated it right now, right? So that's just the nature of it. We have lots of correlations, very few causation studies. Although, again, in mouse models, I don't know how much faith we do have more there. And then here's another thing. It may be lots of genes, but also just may be lots of different biological defects. Maybe it's a combination of genes and microbiome and something else we haven't even discovered yet. So there's a lot yet to learn. All right. So that's what I have for you about autism, and thanks for listening.